Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion and I'll ask our speaker your questions. Your questions in the Q&A window will only be visible to myself and our speaker. So thank you for joining us today. It's now my pleasure to introduce our speaker, Zheng Wei, PhD, Principal Scientist, Group Leader, Downstream Processing Group, Bioprocessing Technology Institute, Agency for Science, Technology, and Research.
Thank you for the introduction. Today, I will be speaking about a polishing strategy for removing impurities in bispecific antibody purification. As we know, bispecific antibody, BSAB in short, is an artificial man-made protein that can simultaneously bind to two different types of antigens or bind to two different epitopes. By doing so, BSAB can form two different drug target interfaces, either enriching drug at relevant sites of action or linking drug to a specific biological effector. Currently, there are 13 BSAB drugs available on the market, and 10 of them were approved in the past 2 years alone. Due to its unique dual targeting capability, BSAB can potentially provide huge therapeutic advantages over traditional monoclonal antibodies, such as BSAB can give higher binding ability to target, can boost cytotoxic effect by directly recruiting effector cells, can effectively prevent drug resistance by simultaneously blocking two different immunopathways, can decrease side effect due to the higher binding specificity. At the same time, BSAB also can potentially generate new functionalities that are not present in any combination of parental monoclonal antibodies. Although BSAB can potentially provide huge therapeutic advantages, but at the same time, it also poses big and unique challenges in downstream processing. In a nutshell, the major challenges in BSAB purification are the byproduct and impurity profile is complicated. Besides the traditional process-related impurity, such as whole cell protein and whole cell DNA, BSAB also can generate a variety of product-related impurity, including mispaired species, a variety of antibody fragments, and also high level of aggregate. Especially, the mispaired species are normally close to our BSAB target in terms of their physical chemical properties, thus being challenging to be reduced. The level of byproduct and impurity is normally high. Moreover, many BSAB molecules are highly aggregation prone, especially if XCFA domain is utilized as one of the building blocks. Mixed-mode resin may play a major role in BSAB purification due to the high resolution it can offer. One of the notable mixed-mode resin is CHT, ceramic hydroxyepitete medium. CHT comprises two different binding sets, the positively charged calcium C site and the negatively charged phosphate P site. C site primarily bind to carboxyl groups on proteins and phosphoryl residues on DNA through calcium metal affinity, and the product elution can be achieved by increasing phosphate concentration in the solution. P sites primarily bind to ammonia groups on protein by electrostatic interaction, and product elution can be achieved by raising conductivity using any type of salt. Although C set and P sets are two independent binding sets, but they can do work cooperatively under suitable conditions. Calcium C sets bind to carboxyl groups on proteins, phosphoryl residues on DNA by metal affinity, but at the same time, there is also strong repulsion between phosphate groups of P sites and the DNA and the protein due to their same charge. But the addition of high concentration of sodium chloride could weaken this repulsive force, thus leading to the relative strengthening of C-site binding. A similar concept also applies to the interactions between protein ammonin groups and the P-site. In our study, 3 Bispecific molecules were chosen, and both molecule A and B belong to asymmetric format, while molecule C belongs to symmetric Bispecific format. The performance of cis-T resin for Bispecific polishing was evaluated in comparison with a typical cation exchange resin. Both sodium chloride and phosphate gradient elution were performed for cis-T, where only sodium chloride gradient elution was explored for CEX. So 3 Bispecific molecules and 3 conditions, 9 runs were done in total. Result here shows that cis-T chromatography could offer better aggregate removal than CX for all 3 tested Bispecific molecules, especially for CHT sodium chloride elution runs. It can reduce high molecular species to 0.5% each and below, and this can be explained by CHT calcium C-site cooperation. Bispecific are basic proteins. Therefore, their binding to CHT is P-site dominant with some C-site cooperation. During CHT sodium chloride gradient elution, higher conductivity triggered protein elution, but at the same time, high sodium chloride concentration also strengthened affinity between C-sites and carboxyl groups on protein surface. Since aggregates were larger than monomers, this C-site retention effect was stronger towards aggregates than monomers, and this difference in C-site cooperation between aggregates and monomers during elution enhanced their separation resolution. Next, let's take a look at low molecular weight species removal. For molecule A and B, CX showed better LMW removal initially, but LMW removal could be improved by adding a optimized post-load wash. For molecule C, CHT phosphate gradient elution provided a biased LMW removal. After one-step CHT polishing, all eluate purity reached 97% and above. Let's have a closer look at LMW removal by CHT. CHT was able to remove a variety of product-related impurities, including the knob and HO homodimer, Bispecific monomer lose 1 or lose 2 lead chains. Let's move on to take a look at process-related impurities, including whole cell protein and whole cell DNA. Where whole cell DNA was almost completely removed in all runs tested, CHT sodium chloride gradient elution runs provided a biased whole cell protein removal to below 100 ppm. Again, this can be explained by C-site cooperation. As we mentioned just now, Bispecific are basic proteins. their binding to cyst is P-site dominant, where the majority of wholesale proteins are acidic proteins, so their binding to cyst is C-site dominant. During sodium chloride gradient elution, the increase of sodium chloride concentration was only able to abolish the P-site binding, but not the C-site binding. Therefore, only our target BSAB were eluted out, where wholesale proteins are still bound to the C site. Another big advantage of utilizing cyst resin for BSAB polishing is that cyst can significantly mitigate chromatography-induced aggregation. In our study, we calculated the ratio of high mercury species content in all elution fractions over high mercury species contents in the load. The high ratio here means more aggregate formed during column runs. From this graph, we can tell that cyst can significantly mitigate chromatography-induced aggregation, especially for highly aggregation-prone molecule C, which contains two SCFV domains. A possible mechanistic explanation for the minimum aggregate formation by cyst is likely due to the presence of calcium ion at C site. Cheotropic salts such as calcium chloride have been commonly utilized as an additive in protein purification, especially in monoclonal antibody purification to suppress the formation of aggregate. And the calcium ion on cyst resin here may provide a similar effect. In summary, 3 BSAB model molecules were polished by one-step cyst resin to over 97% purity. Cyst provided efficient removal for both product and process-related impurity. And also, cyst minimized chromatography-induced aggregation for aggregation-prone BSAB molecules. And this may be due to the C-site cooperation of cyst. Here is the end of my today's talk. I would like to take this opportunity to thank all my team members in downstream processing group in BTI, especially Dr. Nata, Ms. Wang Xinhui, and Ms. Liu Jiamin, who did all the work for today's talk. And also my colleague Dr. Yang Yuansheng from Cell Line Development Group in BTI, who provided all the cultures. And also Dr. Kobayashi-san and Dr. Maria-san from Hoya Technosurgical Corporation, Singapore. Thank you for your time to attend this webcast today. I'm happy to answer any questions you may have. Thank you.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speaker now. So our first question is, why was only sodium chloride gradient elution explored for cation exchange while both sodium chloride and phosphate gradient elution were tested for CHT?
Oh, that is a great question. So for cation exchange, sodium chloride elution is the most commonly used elution strategy. And for CHT, it's a mixed mode resin which comprises two different binding sites, C site and P site. As I mentioned just now during the talk, C site elution can only be achieved by increasing phosphate concentration in the solution, where for P site elution can be accomplished by raising conductivity. So just like cation exchange, therefore sodium chloride elution was investigated as well for safety. Hope this answers your question.
And our next question is, will the current effectiveness of impurities removal by CHT be maintained at a larger scale?
Another very good question. Yeah, currently the results were only obtained from small-scale experiments, and we are going to further optimize and scale up the process soon. But since the process we developed here is a scalable one, so we do believe that CHT is still able to effectively remove process and product-related impurity at a larger scale. Thank you.
All right, well, it looks like we're running a bit short on time, so if we don't get to your question, we'll be passing them along to Wei, who can follow up with you directly. Please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is, what other type of resins were used post-protein A capture?
There are many types of resins have been commonly used as a polishing step for antibody purification post-protein A capture, such as cation exchange, anion exchange, hydrophobic interaction, and mixed mode, etc. But when it comes to bispecific antibody BSAB polishing, as many BSAB molecules are highly aggregation prone, so some commonly used polishing strategies such as especially bent loop mode purification methods could potentially cause severe aggregation of column-induced aggregation, such as cation exchange, mixed mode, and even protein A capture itself. And this phenomenon has been reported in several papers already. So that's why I mentioned during my talk that it is beneficial to use CHT as it can mitigate the potential aggregation formation on column. Thank you.
Well, thank you, Wei, and thanks to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.